In links, several projects are using induced pluripotent stem cells, or iPSCs. In this lecture, I will briefly discuss what are iPSCs, for those of you who are not familiar. Before we were able to make iPSCs, there was much debate about using human stem cells in research because the only place we could obtain them was from a developing embryo. But before we continue, we need to define stem cells. Stem cells have few properties that make them unique, and potentially very useful for medical research and therapeutic applications. The unique properties are that these cells can self-renew forever, so they don't age. The cells can differentiate into all adult cell types, and these cells can be injected into a developing embryo and can contribute to the formation of all tissue types, so they can create an entire organism. In 2006, Takahashi and Yamanaka published the first evidence of our ability to make iPSCs. These findings earned them the Nobel Prize in 2012. This was a huge breakthrough because we now can make stem cells that are indistinguishable from embryonic stem cells from adult cells. So here I created some animation that describe the process of making iPS cells. The process typically starts with a skin biopsy. The goal is to obtain skin fibroblast cells as a starting material. These are the adult cells that will be converted to stem cells. Using a virus envelope, the cells are infected with gene construct that drive the expression of 4 key genes: OCT4, MYC, SOX2, and KLF4. The virus envelope transfection method was the initial method, but now this is done differently to avoid integration of the transgenes into the cell's DNA. These 4 genes, OCT4, MYC, SOX2, and KLF4, are transcription factors. This means that their protein product binds to the DNA to regulate the expression of many other genes. Culturing the infected fibroblast cells in the right conditions, after about 15 to 20 days, colonies of stem cells start to appear in the dish. These can be isolated, and experiments show that these cells are almost indistinguishable from embryonic stem cells. Once you have stem cells from an individual person, you can differentiate these cells into different types of adult somatic cells, such as neuron, myocytes, which are heart cells, or leukocytes, which are red blood cells. You can now study these cells to better understand disease mechanisms in an individual. This is what is done by Neuralinks to study ALS, or to understand how human cells respond to drugs, and this is what is done by the Detox Link Center. One hope for iPSCs is that they will be used to treat diseases that would benefit from cell transplantation. Diseases such as diabetes or Parkinson's are some obvious candidates. And you can read more about stem cells and iPSCs. It's a very large area of research, and here we just gave you an introduction for those of you who are not coming with a background in this field.